All right, so like I said, this is lesson 16.1, and it is looking at selective breeding. Now, this last chunk is all about biotechnology, but before we got into the bio of the technology or the technology of biotechnology, people were doing this already with organisms for years and years and years, thousands of years. Thousands of years, we've already kind of been breeding and selecting for certain traits. And so that's what we're going to take a look at today is how have we selected for certain traits and what exactly do we do or how exactly do we do those selections? How do we go about picking things? So we're going to talk about changing the living world. Now, you guys can make small changes to your own little world, right? You can clean up after yourselves. You can, rearrange your bedroom maybe. But we're talking about the living world. So changing how things live, where things live, and what happens. How many of you guys plant gardens? Any garden planters here? Okay, about half dozen hands or so going up. In gardening, you are kind of doing a little bit of this, a little bit of changing the living world. And you might be thinking to yourself, well, not really. All I'm doing is planting a garden. Yes and no. I mean, the seeds you choose to plant for your garden, how picky are you on those seeds? Do you look at different types of peas? Do you look at different types of tomatoes? We have a little salsa garden in our house, and we plant different types of tomatoes and peppers to try to maximize our type of salsa. So normally you get some smaller tomatoes, they're a little bit sweeter. And we get some big chunky tomatoes that will grow to kind of make up the mass of the salsa. And so us picking the different kinds of tomatoes kind of goes into how we're going to make that stuff. And that's what's going to happen, or that's what we're talking about in this lesson is why are there different types of apples? Why are there different types of tomatoes? We're going to look at how we use selective breeding to do these things, how to kind of bring about different types of organisms, and then how can we increase genetic variation. So in selective breeding, we're working to kind of reduce and pinpoint certain traits, whereas in increasing genetic variation, we want a variety. We want other stuff. We want to be able to breed in different traits or bring in different traits that might be beneficial. How can we do that? How can we do that? All right, take a look at this picture here. What do you see? Lots of dogs, right? Lots of dogs. Can anybody name these breeds? I'm sure my daughter can name these breeds. She is such a dog fanatic. I love it. But can we tell, are these certain kinds of breeds of dog? Anyone see one here they recognize? This German Shepherd right here, okay. Labradoodle, this guy in the end, is it a Labradoodle? Okay. A couple Spaniels, that's these guys here, right? Boston Terrier, is that what this guy here? No. These guys. Okay. What about this thing? It's kind of a mix. Looks like a little bit like a sheepdog and some other stuff kind of mixed in there. This little guy here, kind of like a beagle looking kind of thing. Pardon? Yeah, like a beagle mix. So you can see there's a variety of different kinds of dogs here. Well, how did these dogs get to be so different? Selective breeding, exactly. What is the difference, or what is the purpose that people would have bred to get the traits you see in this big old German Shepherd? What was the purpose for breeding a dog like this? Jedi? Herding sheep, kind of helping corral and move animals around. And if you've ever seen German Shepherds, they follow commands pretty well. When we grew up on the farm, when I was a kid, we had a couple German Shepherds. When I was a real young kid, my dad had a dog. His name was Fritz. And Fritz was pretty good. He could like get the cows in. He could do a lot of good things. We had a second and third German Shepherd, Spike and Spike 2. Spike made some bad choices, so Spike had to be let go. But Spike 2 was awesome. Okay? So these German Shepherds are really kind of cool dogs in the fact that they were bred to do certain traits. What about these guys down here? What are these guys bred for? Anybody know? What's the name of this breed again? Type of terrier, right? So what do terriers do? What are they useful for? Yeah, Jocelyn, do you know, or are you just putting your hand up? No? A little bit.
They're like hunting, aren't they?
They are show dogs now, but they did have a real purpose. They did have a real purpose. They were bred for, do you know?
Hunting, right?
It's a type of hunting. I believe they were bred for hunting like rats, I thought. Small things. Is that right? Olivia's shaking her head. Yeah. Okay, they've got a very muscular front end for digging and kind of getting into dirt. And they've got that short nose so they can kind of try to get rats. They have small ears so their ears can't get attacked and bitten by the rats. But yeah, I think they're kind of bred for hunting rats. What about these guys here? Another type of hunting dog. Notice very long ears. Okay, they probably use their smell to try to help sniff out critters, and their ears kind of help, like a bloodhound, kind of help to sweep up smells and kind of collect those smells for their nose. So you can see a lot of variety. What about this Labradoodle? That's mostly for looking at. Yeah, it just looks nice. And they're nice dogs. They have nice temperament. So selective breeding, we breed organisms that have desired traits. Now, of these dogs here, some of them might make good house dogs. But when you're looking at dogs, you got to kind of keep in mind certain dogs are bred with certain traits in mind. For example, we have two little dogs. One of them is, we think, kind of a cross between a wiener dog and a Chihuahua. His name is Rex. He's a little cutie. He's a good, nice little house dog. Okay, he's medium energy. He's kind of getting a little chonky right now, so we got him on a little bit of a diet. Even though he's little, he's like, we call him our little potato boy. Hey, little potato boy, come here. He look a little potato. He's a little round guy. And then we have Jack, and Jack's a Jack Russell Terrier Chihuahua mix. So he's got like the face and head of a Chihuahua, but like a little bit bigger, like a Jack Russell Terrier. So, great house dogs. I'm not a huge fan of the humongous house dogs. My brother and sister-in-law are. They have two big retrievers that live in the house. I'm just like, these things are humongous. They take up so much space and they leave so much waste behind. And it's like, yeah. So anyways, depending on what you're looking for, you can see there's different traits for different organisms. And how we do this as a collective is we look and take advantage of naturally occurring genetic variation. So over years and years and years, the German Shepherd has been bred to be a good herding dog, following commands, able to be very well trained. Typically, you can train them as service dogs as well. They listen and obey very well. Some of these other dogs may not be very good service dogs, may not be very good at helping out in those capacities. So those traits that we're looking for, that really is biotechnology in selective breeding. But selective breeding happened for years and years and years before we kind of knew about the biotechnology end of it. Okay, let's keep rocking. Here we have two plants. And from the text, if you read that first section here, you read about this plant called, I think it's called Teosinte. It looks like it's how I'd pronounce it, Teosinte, which is said to have been kind of the great, 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 great grandfather of maize, aka corn. How many of you guys enjoy corn on the cob? Looking forward to it in the fall. Usually, early corn's usually up around late June. People have corn already up. But this teosinte and corn, while technically kind of they're related genetically, you can kind of see there's a path to them. What are some differences between these two? What are some differences? Janessa? The color, right? This teosinte kernel, kind of a greenish kernel, whereas corn, very much yellow. Okay, other differences. The size, yeah. It's kind of a little bit hard to tell from this picture, but this teosinte is very small. It's a very small kernel, probably a couple inches. Whereas corn, you guys have probably all seen corn. You know, you can get 6, 7, 8, 9 inch corn cobs. Anything else that's a difference? Yeah, Janessa? The amount of kernels. So here, teosinte, 2, 4, 6, maybe 8 kernels. Whereas corn, I mean, this row right here alone probably has, I would say, 30. And they usually have somewhere between 8 and 12 rows. So that's a lot of corn. That's a lot of kernels. And over time, this teosinte was kind of bred for this trait to grow really big seeds and really big kernels. And as it happened, ancient Mexican culture, ancient Native Americans in Mexico that started growing teosinte and kind of growing it better and better, this natural plant was able to live out in the wild pretty well. But as they domesticated it, that's kind of the name we use here is domesticated. As they domesticated it, it became less and less able to survive on its own, and it needed people to help kind of tend and care for it. It does not do well with weeds. So when weeds come in, corn dies. And farmers here, even now, we use different types of herbicides and different machinery to help clear out the weeds so we don't have weeds growing with our corn because the corn cannot survive in a weedy place. So it's kind of interesting to see how over time these organisms have been changed to feed people. Other ways that we do selective breeding. What is this a picture of? Do you guys know what these are? Potatoes. What's the most common type of potato that we have here in the States? Someone say it. Like your standard white potato, right? What kind of potato is it? It's actually a Burbank russet potato. You heard these words? No? Yes. Some of you have heard these words. Burbank russet potatoes. And it's named Burbank after the guy, Luther Burbank, who did a lot of work with organisms. So what Burbank would do, he would take, so say you've got this potato that grows really nice and big potatoes, and you've got this other potato that's really strong and resistant to disease. So the really good big growing potato, if you cross it with the disease, uh, um, the one that can survive in diseases, you cross those two, you get this Burbank potato. So it's a big, tasty potato that also can survive against different pestilence and different diseases and things like that. So this happened actually kind of in response to the potato famine that happened in Ireland years and years ago, because they had a type of potato that wasn't able to survive against a fungal rot that caused that potato to die. And so through research, other people have developed these other potatoes, and Burbank's potato is kind of like the main one. And it's still a very well-known potato. So that's what's called hybridization. So hybridization is where you're taking a particular quality of one plant and crossing it with a quality of another. We talked yesterday about the pluots, and we liked the softness of the skin, like a plum-like skin, but we also liked the flavor of the apricot. So we crossed those two plants, and it wasn't just like magic. I mean, it was probably hundreds of attempts at crossing it till eventually you got one that actually grew and like did what you wanted to do. Kind of like the video said, even though you're crossing these different apples, it might take up to 5 years before you even know whether that was worth the time of crossing it. So with the pluots, same kind of thing happened here. Burbank potato, another example. Hybridization, taking the best traits from two and putting them together. Another way that we use selective breeding today is called inbreeding. Inbreeding. Inbreeding is where you cross organisms that are of the same family line. Now, in humans, this is strongly frowned upon. This is why you can't marry your cousin. Sorry. But strongly frowned upon because of the fact that there are so many genetic recessive alleles that when you inbreed, there's a high chance that those recessive alleles are going to populate. They're going to show up. And so this is why in organisms like people and animals and stuff, when you start to inbreed a little too much, you wind up getting a lot of genetic problems. However, if you do it right and you're careful about how you're inbreeding, you can actually get a fairly strong species or subspecies. Like, I'm talking about dogs here, okay? Yeah, don't look at me like that. I'm talking about dogs. The way we get the German Shepherds that we have now happen from inbreeding over the course of time. And while we inbreed those dogs, they're able to produce very particular traits. However, if you do that too much, then you wind up releasing those recessive alleles, and then your puppies aren't going to do quite what you want. So there's kind of that fine line of having a good breeding dog and being able to find that good cross. So inbreeding is another example. In plants, it works a little better. In fact, when you talk about like what Mendel did with the pea pods, it was basically inbreeding. I mean, it was like breeding them right back into each other. And so that breeding within each other kind of showed those different traits and pulled those traits out. Same kind of concept. With plants, it works a little better. With organisms like animals and things, it gets a little bit spotty. Okay. All right. Now, the other half. So selective breeding allows us to choose. Increasing variation, that's another option we want to dig into, right? We want to be able to change and increase the variety that we see within organisms. So how do we go about doing that? How can breeders, how can we increase genetic variation within a pool of organisms? Anybody read it? Do you remember from the reading? Olivia, I'm looking towards you. Can you carry us? Not at the moment. It's okay. I put you on the spot there. All right, one way is bacterial mutations. So as we mentioned back in chapter 15, when we have those idea with the plasmids and we used our restriction enzymes and we were able to snip apart different genomes and we were able to grab certain genes and put them into stuff, well, we can do that to a degree with organisms that are multicellular to a degree. It's not perfect by any means, but we're kind of getting closer. So what does that mean? We take mutations and we're able to push them in using bacteria into other organisms. For example, the beginning of the chapter, there is a picture of a glowing fish. That fish has a particular protein grafted into its cells that allows it to glow. And you might see these fish when you go like to Walmart or to the fish store or PetSmart or whatever, and look at the fish. Some of those fish literally are glowing. And that is because of a genetic mutation that we use bacteria to help insert into those fish. Isn't that wild? We made those fish. Those are genetically modded fish. And they have that particular trait. Another one is polyploidy in plants. That's another way you can increase genetic variation. So when we have certain things like watermelons that have seeds, naturally when they're 2N, if we make them triploid or triploid by making them 3N, now the seeds go bye-bye. And we just get the tastiness of the watermelon. Now, granted, I'm guessing those early ones that they were doing the triploid with didn't turn out so good. And so they kept trying it again and trying it again and trying it again until they got a subspecies, if you will, of watermelon that's pretty tasty and maybe can grow well and grow fast and grow in certain conditions. So all those different pieces provide for additional genetic variation. Here are some examples of polyploidy that we have. So you can see the regular domestic oats. So growing up in the farm, we would do oats. We usually did barley more often than we did oats, but we do oats. The normal haploid number is typically 7, but the chromosome number, 42, because it's a 6N. So if haploid is like half, it's like how many is in a gamete. So if you take 7 times 6, you get 42. That's how they got the 42. You guys catch me on that? So by using polyploidy techniques, we can increase the chromosomes, thereby making the oats bigger. Because what happens when there's extra chromosomes in plants? Not all the time, but a lot of the time, it actually makes the fruit bigger. Bananas are way bigger than they grow naturally. Natural bananas are only like a couple inches. They're tiny. But the bananas you get from the store, those big bunches of bananas, they are typically triploid or quadruploid. Or they're 2N. I don't know if they're quadruploid, maybe they're just triploid. So they're 3N, and because of that, they just grow bigger. Kind of wild. So pretty neat stuff. Again, if you didn't do the reading, make sure you check into that reading. So quick review to close up here before the review questions. How do we give me an example of what I just talked about, of how we use hybridization to increase or improve plants. Give me one example. I gave you about 6. Show me you're paying attention. Trin. Watermelons, okay? We use hybridization maybe in water. I would say that's more along the polyploidy piece, but we can hybridize watermelons to do different things, to like be stronger or be better, right? Yes, Liv? Pluots. Yeah, that's another good example because we're crossing and getting the traits of a plum and an apricot. Apples. Lots of different kinds of apples. All those, like, what does the video say? Like 3,500 or 7,500 different kinds of apples. Lots of kinds. That's all done through hybridization. What about inbreeding? How does inbreeding relate to dogs? Dogs, Jocelyn. How does inbreeding relate to dogs? Yes, Janessa. Okay, Haley. That's how you get purebreds, basically. When you take 2 dogs that are of the same bloodline and cross them, you will then continue to keep those chromosomes very tightly connected. And so the litter that you get, the pups you get, will be very similar to the original traits. And you can kind of do that to a certain degree. If you do too much of it, it gets a little, you start having those recessives show up. But for the most part, that's kind of how they get different purebreds. And the thing is, if you're into dog showing, I'm not, but if you're into that, you probably are able to trace back the bloodline of your dog. And that's kind of an important piece because if you can trace back your bloodline, then you can use your dog as a breeder to be able to breed with others. Haley, got more to share? Yes. Yep. It's more expensive, but that's the thing is if you're planning to be a breeder for this particular breed, you have to have proof. And then usually the pups go for a little bit more dollars because you've got the paperwork to kind of prove the bloodline. So All right, very good. We have three review questions. I'll post the assignment here shortly. We're going to go through 16.2 tomorrow. Explain the process of selective breeding. Why would breeders want to introduce mutations into a population? And then how do biotechnologies like hybridization and the introduction of mutations, how do they increase genetic variance? Okay, hopefully you can answer those three questions after today's lecture. So thanks. And like I said, we'll talk about 16.2 tomorrow. So if you didn't read that,